All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So I enjoyed the talk with Dr. Eli Schwartz from Israel this morning. And today, this was another topic that I think is really important. The reason for that is there is so much confusion. I had the confusions as well. And so it is important once for all to look at the science behind a spike protein produced by a vaccine. And that's about it. We should talk about it. We should know what it is, and then we'll continue from there. So let's start our discussion. So this is our little stable spike protein gifts. And let me show you the references. So this is drbeen.com. This is just a simple table that the amino acids are little bricks that are used to make our genetic material. So amino acids can have single letter names. And this is useful for us because, as I discussed the mutations in the spike protein, some of these single letters may be useful. So I thought I would share this link with us. It is in the description as well. This is one more link here, and I have used parts of this study. This study is viral targets for vaccines against COVID-19. Then this is another study which talks about SARS-CoV-2 messenger RNA vaccine design enabled by prototype pathogen preparedness, which is what they mean is that in 2015 or even before, there was some vaccine designs that were researched upon, and now those designs are used. Then this is a thermostable closed SARS-CoV-2 spike protein trimer. And then finally here, conformational dynamics. So I'm going to now take these one out. They churn my computer and cause a little difficulty in presentation. So I'm just going to leave this one here. This one. And let's start our discussion. So the basic discussion is the following. Will a spike protein, which is generated by a vaccine, cause cytotoxicity or damage because it will bind with ACE2? So the short answer is no, it will not. Second part of the answer is that the damage can occur because of the immune system. So when you have a vaccine injected in a deltoid muscle, for example, and there is local reaction, that is local damage because the immune system is responding and releasing cytokines. The cytokines also cause pain. So it's not necessary that every pain is damage. Cytokines cause pain as well. That is an alarm for us to say there is something going on here. Then when the systemic reaction occurs, for example, fatigue or fever or myalgias or tiredness or sleepiness, those are the signs of, again, the immune system and the cytokines that are being released. So, of course, those are the responses to the vaccine and the spike protein. And in that response, there can be cell damage as well. So spike protein itself, by binding to ACE2, no damage. The spike protein, by activating the immune system, can cause local tissue damage. With this, what I want to explain is, why does the spike protein not cause a damage? So the theory that the spike protein will be in the blood vessels and go and bind with the ACE2 and cause endothelial damage, that is not true. Similarly, the theory that it is going to go and bind with other cells and cause damage, that is also not true. So let's see why not. So here in summary, spike proteins are generated by vaccines that are stabilized in non-fusible state. So for this summary and for this understanding, please realize that spike protein are in two states. If you look at my hand, the spike protein can be in a closed state. That is how the virus, when it enters our body and is present wherever it is, in the tissues, in the throat, in the lungs, the spike proteins of the virus are closed. And then when it binds with the ACE2, then the spike protein opens up. So imagine that this is called, the closed state is called pre-fused state. That the virus is not fused yet, and before fusing, this is the state of the spike protein. Then when the spike protein binds with the ACE2 and it opens up, this is called post-fused state. And the question is that when we are building vaccines, what state is the best for the vaccine to work on? And the pre-fused state is the best. So when a vaccine is supposed to produce a spike protein, it is going to try to create a spike protein that is stable in prefused state. So immune system has a target which looks like the viral spike protein in the closed state and work with that. This also means that if the spike protein is in a closed state, it cannot really cause any binding with the ACE2 and damage. So that is a summary. Let's look at the mechanism for why this happens, or how does this happen. So imagine here we have a virus. Virus has the SARS-CoV-2 has many proteins. That some proteins that are of interest to us are the spike proteins that we are very famously aware of. Matrix protein or M protein, which is also part of the virus, and we make antibodies against that as well if a vaccine has it, for example, inactivated virus. Envelope proteins, these are also some proteins that are present in the membrane or the envelope of the virus. Nucleocapsid protein, which is a protein called N protein. It is present inside the virus and it wraps the RNA part of the virus. It kind of provides a structure around which the RNA is wrapped. So these are the main proteins. We are going to talk about the spike protein because that is the target of the vaccines as well. And keep in mind throughout this discussion that spike protein is in two states: closed state or pre-fused state, and open state or post-fused state. The target, the objective of a vaccine is to create spike proteins that are in pre-fused state, not because they think that this is going to be somehow damaging the system, although this is a good outcome, but because they want to create a spike protein that looks to immune system as close to the spike protein as it is on the SARS-CoV-2 when it is present in our system and is not bound yet. So here is a spike protein, and let's look at this structure one by one. This spike protein here has an S1 domain. So if I go here for a second, this one in yellow is the S1 domain. It's a trimeric protein. It is homotrimeric. That means all of the spike proteins are similar and they are in this trimeric structure. So what is important for us here is to realize that there is an S1 part. Then this blue part is the S2 part. And then this red thing down here is the virus. So on the surface of the virus, we have a spike protein that has two major components, S1 and S2. We know about that forever. Now, if I go back here for a second, the S1 component has an N-terminal end. What does that mean? If you take a rope, so let's look at my hand, and there is a pen in my hand, although blackish, so I don't know if you can clearly see it or not. But let's say this end and this end, they are the two ends of the rope. One end is called N-terminal, there is nitrogen attached there, and the other one is called carboxy terminal or C-terminal, there is carboxy. So imagine one end and the other end. So this is the N end. This is one part of the rope. N-terminal is connected with receptor binding domain. This is the part of the spike protein that binds with the ACE2 enzyme. On the receptor binding domain, there is a specific part that actually connects with the ACE2 that is called receptor binding motif in a closed state. So imagine if I am a spike protein. I'm gonna show myself as a spike protein for a second. Imagine I am a spike protein. This spike protein has this S1 unit. My hand is S1, and I'm going to put it on my head. So usually the S1 is what is showing when the virus is not bound to anything. So then when the virus, sorry about this little demonstration, when the virus binds with the ACE2, then what happens is our TMPRSS2 protein, remember the proteolytic protein on the surface of our cells, TMPRSS2, that TMPRSS2 cuts down the S1 unit from S2. So what happens is S1 unit is moved away and cleaved and cut away. What is left now is the S2. And S2 on its surface has a drilling machine that binds with the cell membrane and fuses with it. So this is a very important concept. I'm going to repeat a part of it. Now, if we look at this structure again, now I can hopefully explain on the structure. So if I go back here, imagine this is the S1 unit. This unit is covering a lot of part of the S2. And if you see here, this is a drilling part of S2 or fusion protein part of this S2. And S1 has kind of kept it at bay, kept it closed. Now, S1 unit then connects with S2 unit, and first connection from receptor binding domain is to the fusion protein. Fusion protein is the one that will fuse with the cell membrane to allow the virus to get into our system, our cell. Fusion protein is then connected to some other proteins that are here. And finally, there are two big chunks of proteins as well. This whole unit is called S2. But please remember, S2 does not have a chance to do its function until S1 is removed. This is like you remove the cap of something and then the actual razors come out. So how does it work then? What happens is, if you look here, when a virus binds, I'm going to increase the size here. When the S1 unit binds with the ACE2, we know that this little silly TMPRSS2, our little cute protein on the surface of our cells, TMPRSS2, it will cleave, it will cut the rope between S1 and S2. That cleavage or that cutting is done between S1 and S2 and then within S2 as well. The end result is that this S1 part will float away. It will be dissociated. And what will be left is the S2 part, and S2 part is going to open up like a weapon and like a little drill machine thing and get stuck with our cell membrane. So the ACE2 binding part is broken away and removed. What is then revealed is the weapon. That weapon is the S2 part. So how does that weapon work? If you see here, in this diagram, for your viewing pleasures, I was actually drawing this. I enjoy drawing. So this is like a little kid who is just drawing, and I enjoyed it. So if you see here, this is the S1 unit. It has been disconnected from the S2 unit. And if you see the S2 unit, now it's changed. It has become a straightened spring-like structure. Let's go back for a second to see here. Over here, the S2 unit is all this coily thingy. But as soon as the S1 unit is removed from it by our TMPRSS2, we have a traitor in our cells. Well, that's a nice, cute protein. It thought it's doing its function. As soon as the S1 is removed, the remaining S2 opened up, unleashed, just like a clasp knife, and you open the knife with the blade that comes out. And there is a fusion protein attached to it with the rest of the things. This fusion protein will get connected to our cell membrane, and the rest of the function is of this fusion protein to get the virus into the cell. The S1 unit, the receptor binding domain, they're all useless, they're trash, they're thrown away. So then what happens? We have a problem. If we humans, scientists, researchers, make this S protein through genetic material, and we take that genetic material from the virus itself, the S proteins that are formed, if you just drop them in our body, they would automatically open up. So imagine that we have a knife that closes, but if you just put it on the table, it would automatically open up. And you clasp it again and you put it on the table, it opens up again. The S protein by itself, when present at our natural normal pH, it opens up automatically. Even when it is not bound to ACE2, it just opens up. That is the post-fusion state. This is why we say that S protein, when artificially generated, is metastable. Metastable protein would automatically open, and we have a problem. What is the problem? The problem is actually automatically open spike protein is not a big problem in terms of binding with ACE2 because its pieces have just floated away, broken down. The problem is that our immune system, when it recognizes an open S protein, an open knife, it might react to the blade of the knife, which when the virus will come in, that will have that blade closed. So our immune system will not even know that the virus is in here because virus's S protein is going to be in a closed state and the blade is hidden. So then for researchers, the problem becomes, how do we make an S protein that does not open so that our immune system recognizes it? And this is a research that was done in 2013, 14, 15. It's not a new research. And the folks who did that research, their research is used till to date. So I hope that we are on the same page, that the objective now, if you and I are making a vaccine, our objective is to sit down and think about how do we make an S protein that does not open up. That's it. That's the objective. So let's see, what do we do? What we do is, imagine that knife. I'm giving the analogy of the knife. Let's continue with that analogy. So let's say this is the knife. And this knife has the blade that is in it, right? So let's say this is the blade that is clasped in it. Imagine if we wrap this knife in a couple of ropes or ties. Now, even if the knife is present, even if it has a spring to open it, it cannot open because we have tied it down. That is the objective. How do we do that? Here is how we do it. Very beautiful mechanism. I loved it. How do we make a spike protein that does not hurt us, but still allows the immune system to become trained? Here is what we do. Number one, the receptor binding domain can actually be repeated. This is nothing but a genetic material. So let's say there is a paragraph of some text. You just copy paste that paragraph 3 times, and now we have 3 receptor binding domains. So this is a dimer or a trimer of the receptor binding domain created. Why? So let's say if I go back to that analogy of my hand here being the receptor binding domain, you put 2 hands there for the virus, for the immune system to recognize. Virus doesn't have 2 hands, but immune system now has more epitopes for the receptor binding domain, so that immune system can be trained more on the receptor binding domain. So the vaccine can actually have dimer or trimer of the receptor binding domain. And let me quickly show you. If you see here, this is the receptor binding domain dimer, and this is receptor binding domain trimer. And if you read here, immunogenicity enhancement, that we want the immune system to attack the receptor binding domain. And by showing multiple receptor binding domains on the spike protein, we increase the chance for immune system to bind with them. So that is one change that can be done. This is one. Beautiful mechanism, smart. I sometimes think that, of course, I did not discuss this way. I had been talking about soluble spike proteins and wherever, and the people who generated these research workers, they might be just sitting somewhere saying, guys, don't you know that they would not become active? Okay, so this is one. Second piece. So if you see here, this part, within the S2 subunit, there is this part here of protein where what they do is they create 2 mutations. And these are the 2 mutations here. What are the 2 mutations? One is at the place 986th amino acid, lysine is replaced with proline. Lysine is replaced with proline as an amino acid. So at the place of 900, imagine you have a long rope with beads in it. The bead number 986 was made up of the brick lysine or material lysine. You replace that bead with proline. Similarly, the next bead, which is the place number 987, that was made up of valine material, some nice gem, beautiful, shiny thing, valine. You replace that with proline as well. So when you replace these two, then this knife cannot open. So imagine that you have actually stuck something inside the knife. And now the knife, even if can open, it cannot open anymore because you've stuck it. So there is this mutation done which makes the spike protein get stuck in this state and it cannot open anymore. Then there is another little hook or a claw that is attached to the bottom of the S2 unit so that the three trimers that are present cannot open anymore. So imagine that this part was actually going to unfold. But what you did was you wrapped that as well in a protein, and now the unfolding cannot occur. This is called a trimerization tag. So there is a trimerization tag that is attached here, which causes it to hold in that state. This is very similar to the packaging we see with the materials. Sometimes you buy a mouse or a keyboard or other things, and there are various packings on them so that they cannot open up until you remove the packing and you open it. That is the same thing that is happening. So there is one mutation here, which is 2-proline mutation. This spike protein is called S2P mutated spike protein. This cannot open, this cannot bind. And then it is also trimerized, stabilized, meaning it is stuck in the trimer state, right? These three changes are done in majority of the vaccines, and the examples are Moderna vaccine is this way, Pfizer vaccine is this way, and the Oxford vaccine is this way. Then there are one more change here, which is beautiful. So this change is, remember our protease TMPRSS2, the protein, the cute protein on our cell surface, which is going to cut this area. Imagine if we defeat the TMPRSS2 from cutting the spike protein and opening it. How do we do that? So Johnson & Johnson and Novavax vaccine have this. They have another mutation in this region. Which region? The region that is between S1 and S2. The region where TMPRSS2 cuts the protein. There is another mutation there, so TMPRSS2 cannot reach in anymore and cut it. So now if it cannot cut it, that means the receptor binding domain is going to stay stuck on the spike protein. Spike protein cannot open up anymore. Spike protein is stuck in this state with many mutations, tiny mutations, but many mutations. This spike protein is now very useful for the immune system because the immune system can look at it and be trained, and this training will produce an effect of the immune system that would target the actual virus's closed spike protein, which is the majority of the spike protein state. So I hope this discussion makes sense. I'm going to stop here and we'll go. This doesn't mean that the vaccine does not cause side effects. Vaccine still causes side effects because it causes the immune system to activate. Then vaccines have substances or adjuvants that allow the immune system to become activated as well. And that all thing together can cause side effects. So with this, please do me a favor. I'm seeing some questions. So let's say I'll answer a couple of questions and then maybe we can do a chit chat. Nick says, can we rule out the possibility that a free pre-fusion S protein will target receptors other than ACE2 or have some biochemical hormonal action elsewhere? Thanks. So it is not known, and at least definitely not known to me. So I'm sure that these tests and trials have looked into those things, that what are the effects that a spike protein produces. I do not know that it can bind somewhere else. Spike protein is supposed to bind on ACE2 and nowhere else so far from the virus infections that we know. Good question though. So Roman says, Center for Disease Creation delayed their emergency meeting again due to new holiday. They really care for, yes, Roman, I become frustrated with, they are just, McGarnett says, Dr. Drbeen is I love illustrating, especially when I am illustrating, I'm thinking, how would you see it, and would it make sense to you or not? So it's kind of trying to communicate. Okay, so a couple of people-related talks. Number one, I spoke with Margaret today as well. Her phone is still stuck in a bad state. She went to Best Buy to get it fixed, but she said after sitting there for an hour, they said, now it is time for us to close, so we can't help you. So she was going to go and get it fixed once more. It looks like it's a long process that she would have to send it to Apple, and Apple would do something and send it back. So that is Margaret. What I do is I call her daily to kind of update her. John Snyder, I wanted to say, John Snyder, thank you. Someone, a cool bean, sent a message that John Snyder has been very, very helpful during these discussions as well. So shout out to John Snyder. I usually have very less chance of looking at the talks because I'm kind of focused and stuck there. So John Snyder, thank you very much. And then I heard from France as well that she said, I am a little busy with some personal matters and taking a tiny break. So these are some. I haven't seen Jim. I'm worried about Jim. And Jana today, I think she had her surgery yesterday. I hope she's recovering and she's well. So with this, let's just answer a couple of more questions and then we start a chit chat. So Arun says, how selectively these changes are made in spikes? Arun, do you mean that? So elaborate a little more. Anthony says, thank you, Dr. Bean. Can you comment on cell mitosis with spike protein in them? What would happen? So Anthony, wait for a day or so. I am actually talking with a researcher who does research on protein duplications in the synthesis phase of a cell division. And I'm trying to understand what kind of behavior the cell would show. So I'll update later on. Samina, thank you very much. I've been looking for you for some days. I hope you are doing fine. Eric Dog says, can I put your still drawing on a t-shirt with Bean's lectures? Absolutely. So Eric, I love your comments so much. Your recent comments on the YouTube or in the other places, they just crack me up. So thank you very much for, just like I'm trying to serve, I sometimes feel that some cool beans rise up and they kind of try to reciprocate. And that doesn't mean people have to reciprocate. What I mean is I receive benefit back as well. So your comments always crack me up. So yes, let's see each other in the chit chat and we'll go from there. Please hold on to your questions. Let's start again in 5 minutes and talk more. Thank you, and I'm going to see you soon. Please like, subscribe, and share, and please support this work. There are 3 links in the description. If you can support it, one is for the PayPal, one is for the coffees, and one is to be a patron. Thank you very much. See you soon.